Hey guys, today we're going to continue our unit on DNA and protein synthesis by taking a look at a new molecule called RNA and examine its role in making proteins. This image is showing the double helix molecule of DNA that you're all familiar with being separated and our new molecule RNA being formed from it. This is actually the first step of protein synthesis, but more on that later. As we established earlier, genes are the coding sections of DNA that control the production of proteins. We are made up of about 22,000 genes, and it's these variations of genes in our DNA and ultimately the proteins that are made that make us all unique. These genetic messages can be decoded by copying part of the nucleotide sequence from DNA to RNA. As a result of this copying process, RNA also contains coded information for making proteins. You can think of DNA as a master set of instructions that make you who you are. Sort of like a foreman that has the master blueprints to build a house. Because he doesn't want to damage his master copy, he makes photocopies and distributes it to the rest of the workers. RNAs are disposable photocopies of DNA that will be used to build the proteins. So what is it made of? What does it look like? Like DNA, RNA is a nucleic acid. It is a long chain of nucleotides, which you recall is made up of a sugar, a phosphate group, and a nitrogenous base. There are some differences between DNA and RNA, however. The sugar in RNA is ribose, as opposed to DNA's deoxyribose. RNA is single-stranded, while DNA is double-stranded. And finally, the base uracil is used in RNA instead of thymine. Let's take a look at the actors involved in the production of proteins. There are three main types of RNA, and we will look at what each one does and what they look like. The three main types are messenger RNA, ribosomal RNA, and transfer RNA. First up is messenger RNA, or mRNA. Feel free to use the abbreviation when you take notes. Its job is to carry copies of instructions for assembling amino acids into proteins. Remember, it's the photocopy of our master blueprint. Also take note that it's a single-stranded molecule and that we've got the base uracil here instead of thymine. Next is ribosomal RNA, or rRNA. If you recall, in Unit 1, in the parts of the cell, we often drew ribosomes with little balls. If you were to zoom in far enough, you'd see strands of rRNA wrapped around proteins making a complex. This is a site where your proteins are actually built. Finally, transfer RNA, or tRNA. Notice it kind of looks like a T to help you remember its general structure. Its job is to transfer, again, note the T. It moves amino acids floating around the cytoplasm to the ribosome. Think of these molecules like the construction workers building the house. So these are the major molecules involved in building the proteins. Let's see how we actually build the protein. Protein synthesis is a two-step process. The first step is called transcription. This is where you copy a section of DNA into a complementary sequence of RNA. This takes place in the nucleus. Again, remember, the foreman doesn't want to ruin his blueprint for the house, and so he keeps it locked up safe in the nucleus. To transcribe, you need the help of the enzyme RNA polymerase. During transcription, RNA polymerase binds to DNA and separates the DNA double helix. Once separated, RNA polymerase uses one strand as a template from which the nucleotides are assembled into a strand of RNA. So here's transcription. Notice the DNA double helix has been separated by RNA polymerase. Once bound, RNA polymerase slides along one of the DNA strands and begins attaching nucleotides to it to form the RNA copy. We know that this brown strand is RNA because it is single-stranded, and if you look at the key, you notice that the orange color here, uracil, is present instead of thymine. Again, the cell knows which nucleotide to bring in by following the rules of base pairing which we covered in our study of DNA. For instance, if the DNA template has an adenine, so for instance here is your adenine, here this green, it's going to pair with a uracil. Additionally, if the DNA has a guanine, its complementary base will be a cytosine. So to recap, RNA polymerase slides along a single strand of DNA, adding nucleotide bases to build your single-stranded mRNA molecule. This is going to keep happening until it reaches the stop sequence contained within the DNA. Scientists noticed that the transcription is very precise and predictable. As a result, figured out if they knew the sequence of the DNA, they could predict the amino acids that you could create from it. In other words, the genetic code is like a language of mRNA instructions. We have 26 letters in our alphabet. The genetic code has 4, A, U, C, and G, or adenine, uracil, cytosine, guanine. It's these letters that will form our amino acids, which are like the words of the genetic code. When a cell reads the mRNA, it reads it in three-letter increments called codons. In this picture, AUG is a codon, and AAC is a codon. Each codon codes for a particular amino acid that will be added to the polypeptide chain, the protein chain. It is a very redundant code, and so multiple codons can specify more than one amino acid. This is a good thing because mistakes happen during the copying process, and it's possible that the damage could be negligible if the codon is changed into another that will code for the exact same amino acid. As I said before, the language is so predictable that if we know a DNA strand, we could figure out the complementary mRNA strand. And if we know the mRNA, we can predict the amino acid being coded for. This is a codon wheel, and we will practice using it to determine the amino acids being coded for. To use it, you first need to make sure you're looking at an mRNA codon, not DNA. Once you have a codon, you start in the middle of the wheel and you work your way out. For instance, if your codon was AUG, you find the first letter of your codon in the middle, in this case A, so you're operating within this quadrant. Now again, our codon was AUG, so we found our A, we work our way out to the second ring, we find our U, here it is. Now we're operating within this narrow range of letters. Our third letter was a G, AUG, codes for the amino acid methionine. Now you try. Look up the amino acid for CAG. Are you ready? You should have determined that it was glutamine. Here's your C. We work our way out to our second letter, our A. Here's our third letter, G, glutamine. Notice the redundancy of the code in that CAA could have also given you glutamine. If you didn't quite get it, don't worry, we will continue practicing this in the coming days. So that's transcription, and how we can predict amino acid. Now let's take a look at under the hood at the cellular level to find out how the cell reads mRNA to bring in the amino acids we've been predicting. This is also where our other two types of RNA will enter. Our second step in this two-part process is translation, which is the decoding of mRNA into a protein. You are translating or converting the mRNA sequence into an amino acid chain, and this takes place on the ribosomes. Again, mRNA is transcribed in the nucleus and then enters the cytoplasm where it finds and attaches to a ribosome. Translation begins when the mRNA leaves the nucleus and attaches to a ribosome. As each codon moves through the ribosome, the proper amino acid is brought into the ribosome by tRNA, where it will be attached to the growing polypeptide chain. Each tRNA molecule carries only one kind of amino acid. At the opposite end, there are going to be 3 unpaired bases as well. These bases are called anticodons, and they're actually complementary to the mRNA. They're kind of like ID tags for the cell to know, or the ribosome specifically, to know that it's pairing the correct amino acid with the correct codon. So here's our ribosome, again made up of rRNA and proteins, and it attaches to the mRNA. Notice how the mRNA codon fits into the tRNA anticodon, again following the rules of base pairing and complementary. So we've got A here, adenine, pairing up with uracil, adenine pairing with uracil, in this case cytosine, attaching to a guanine. And it just so happens that this anticodon, UAC, is attached to the amino acid methionine, which, if you recall, in the earlier slides when we looked up AUG, we predicted it was going to be methionine. The ribosome then reads the next codon, UUC, and again, following the rules of base pairing, brings in an AAG anticodon, which in this case codes for phenylalanine. At this point, the ribosome is full, so we need to get rid of some of these tRNA molecules. To make room, our first tRNA lets go of the amino acid and floats away into the cytoplasm, and the ribosome slides along the mRNA until it reaches its next codon, again 3 letters, in this case AAA, 3 adenines, which brings in the complementary UUU tRNA anticodon and its corresponding amino acid, lysine. Again, our ribosome is full, so we'll need to get rid of our second oldest tRNA molecule. And notice in this case, methionine broke off and attached to the phenylalanine. Again, our ribosome is full. This chain here, your methionine and your phenylalanine, will now break off and is going to attach to the lysine. This will keep happening until the ribosome reaches a stop codon, which indicates that the protein is finished and that there is nothing left to translate. At this point, the protein is released and will become modified and folded by other cellular organelles, and at that point it's ready to be used by the body. So yeah, guys, that's transcription and translation. Please come to class with any questions you may have with this material, and we will take the first part of class to kind of review the information, recap what you saw, and kind of clear up any misconceptions. Alright guys, till next time, take care.